My name is Marianela Perez-Torres. I am the Deputy Director for the Division of Chemistry and Toxicology Devices within the Office of In Vitro Diagnostics and Radiological Health at the U.S. Food and Drug Administration, better known as FDA. In this presentation, I will summarize the FDA regulatory oversight over in vitro diagnostic assays, our general perspectives on standardization of clinical laboratory tests, and some initial thoughts on potential paths for regulatory implementation. This is my disclaimer. This presentation is intended for informational purposes only and does not constitute legal or regulatory advice. The FDA classifies medical devices, including in vitro diagnostics, into one of three classes based on the degree of risk they present and the level of controls that are necessary to assure the safety and effectiveness of the device. Device classification depends on the intended use of the device. The class to which a device is assigned determines, among other things, the type of pre-marketing submission or application required for FDA authorization to market. As device classes increases from Class 1 to Class 3, the regulatory controls also increase. Let's start with Class 1 devices. These are considered low risk and are subject to general controls, which are the basic authorities of the medical device amendments that provide the FDA with the means of regulating devices to ensure their safety and effectiveness. General controls include the provisions of the Act pertaining to things like adulteration, misbranding, device registration and listing, pre-market notification, and good manufacturing practices, among others. Most, but not all Class 1 devices are exempted of the pre-market notification procedures. This means that the companies will validate the device and will keep the documentation on file without submitting to FDA. That is unless the assay is considered a Class 1 reserved device or if any of the limitations of the exemptions is met. For example, ALT and the human growth hormone test systems do not need a 510K unless they are intended for point-of-care use, which is a limitation of the exemption. FDA classifies into Class 2 devices with moderate risk, for which general controls alone are insufficient to provide reasonable assurance of safety and effectiveness, and for which there is sufficient information to establish special controls to provide such assurance. Special controls are usually device-specific and may include things like performance standards, post-market surveillance, special labeling requirements, pre-market data requirements, among others. Most, but not all Class 2 devices require a pre-market notification that is known as 510K. The reason why the pre-market notification is commonly referred to as 510K is because it actually refers to Section 510, letter K, of the Federal Food, Drug, and Cosmetic Act. A 510K is considered a marketing clearance application. Therefore, FDA clears 510Ks. The clearance is based on the determination of substantial equivalence. That is basically the demonstration that the new device, compared to a predicate device, has the same intended use and the same technological characteristics, or it has the same intended use and the differences in technological characteristics do not raise different questions about safety and effectiveness. This means that the manufacturer provides evidence to demonstrate that the device to be marketed is at least as safe and effective, or substantially equivalent, to a predicate. And how we define predicate? For FDA, a predicate device is a legally marketed device that has been cleared through the 510K process. There are three types of pre-market notification 510Ks that may be submitted to FDA: traditional 510Ks, specials, and abbreviated. Most of the tests under the purview of the Office of In Vitro Diagnostics, and in fact for CDRH in general, fall under this category. Examples are test systems intended to measure glucose, electrolytes, vitamin D, hormones, among many other devices. Finally, the FDA classifies into Class 3 devices intended to be used in supporting or sustaining human life or preventing impairment of human health, or that may present a potential unreasonable risk of illness or injury, for which general controls and special controls are insufficient to provide reasonable assurance of the safety and effectiveness of a device. Most Class 3 devices required a pre-market approval or PMA. The PMA is the most stringent type of pre-market submissions. Before the FDA approves a PMA, the sponsor must provide valid scientific evidence demonstrating reasonable assurance of safety and effectiveness for the device's intended use. Some IVDs that fall under this classification are assays intended to detect, for example, free PSA, alpha-fetoprotein, HIV, and hepatitis B. Devices that were not known before the date of the Medical Device Amendments of 1976 has not been classified by FDA and are automatically classified into Class 3 regardless of the level of risk for that product. However, in 1997, the de novo path was established and enacted. This is a classification process that uses a risk-based strategy that provides a means to classify novel device types into Class 1 or Class 2. If the de novo is granted, it establishes a new device type. Along with a new classification, FDA will create a new regulation and will establish the necessary controls, like, for example, special controls for a new Class 2 device. If a de novo is granted, the device is eligible to serve as predicate for new medical devices following the 510(k) process. The types of studies typically used to demonstrate substantial equivalence of in vitro diagnostic tests may include, in the majority of cases, analytical studies using clinical samples. This includes studies to assess the imprecision of the assay, the bias or inaccuracy by means of a method comparison, linearity, detection capabilities, specificity. Matrix comparison studies may also be submitted to support adding a sample type that are similar to the sample type used to validate the device. And reference ranges studies are typically included, especially for immunoassays. Each manufacturer should also provide information regarding the metrological traceability of their assays. For some devices, the special controls requires annual standardization by a certifying organization, and I will come back with some examples in the next slides. Depending on the device, some IVDs will require additional studies. For example, the link between the analytical performance and the clinical performance might not be well defined. In these circumstances, clinical information may be warranted. Other studies depend on the composition of the test system. For example, if the new reagent is used with a new instrument that FDA has not cleared before, then electrical safety, software documentation, and electromagnetic compatibility testing should be also included. And again, depending on the device, it may require other studies, like altitude studies, assessment of hematocrit effect, cross-reactivity and hook effect, biocompatibility, sterilization, and the list can keep going on and on. Please note that although this list seems very long, it is not all-inclusive. FDA acknowledges and agrees that standardization or harmonization processes where the accuracy and precision and other analytical performance parameters are improved, maintained, and independently verified in general will be beneficial to patients. And I will share two examples with you. Although standardization of all assays is currently not feasible for analytes, when strong reliable programs have emerged and the clinical community have favored or claimed for standardized tests to ensure patient safety, FDA has found opportunities to implement regulatory requirements that will ensure continued performance of such devices so that they meet the clinical needs of the intended use population. For instance, in 2013, FDA granted a de novo to the first A1C assay that was specifically intended for the diagnosis of diabetes mellitus. For this type of device, because the new intended use raised different questions of safety and effectiveness, FDA was unable to find a device substantially equivalent to a predicate, but work on crafting special controls adequate to ensure safe use of A1C assays that are used to diagnose diabetes. Accuracy and precision of such tests, in part, addressed by the first special controls, and this one requires that the device must have initial and annual standardization verification by a certifying glycohemoglobin standardization organization deemed acceptable by FDA. For A1C assay, one example of the organization that has been deemed acceptable is the National Glycohemoglobin Standardization Program. Another interesting example was our decision for not finding an LCMS assay intended to measure vitamin D to be substantially equivalent to an immunoassay predicate because the technological characteristics of the LCMS device raise different questions that in this case reduce concerns that are specific to immunoassays. That is, the LCMS technology increased the specificity of the assay, thereby mitigating concerns from erroneous results caused by similar compounds known to cross-react and interfere with immunoassays. In addition, the lack of reliance on antibody production and maintenance reduced concerns of assay drift over time. This, in combination with a special control number 1 that requires initial and annual standardization verification by a certifying vitamin D standardization organization, allowed FDA to exempt the device from the pre-market notification procedures, even when it is a Class II device. This is to say that FDA thinks that standardization usually helps. And we have leveraged data from standardization programs to inform our regulatory decisions. And the bottom line message is that as regulatory agency, we don't want to be a barrier or be perceived as a barrier to achieve standardization. The last piece of regulatory knowledge that I think is important for the discussions we will have in December is what FDA requires when a device is modified. In this slide, I'm sharing some of the FDA guidances that provide regulatory advice to manufacturers regarding the requirements for a modification of a medical device. In the next slides, I will focus on the first one, deciding when to submit a 510K for a change to an existing device. This is because most of the IBD devices are Class 2, but wanted to make you aware that Class 3 devices have a different requirement, and as such, we have a separate guidance listed here under bullet number 2. I also wanted to make you aware of the assay migration guidance, which is specific to devices that are subject to PMA and also for certain assays that require a 510K. So the question is, what type of changes will trigger a 510K? And the FDA guidance does not provide a definite answer for all devices, but rather overarching concepts and principles that manufacturers should consider in their decision-making process. The regulatory criteria in 21 CFR 807.81 state that the premarket notification must be submitted specifically when a change or modification in the device could significantly affect the safety or effectiveness of the device. For example, a significant change or modification in design, material, chemical composition, energy source, or manufacturing process. Because the standardization process may cause a drift in patient results and may impact the performance of a test, such change and data to support the modification will need to be submitted to FDA. The level of evidence needed will depend on the modifications made to the assay, which could go from a calibration adjustment to changes in the reagent formulation to meet standardization program criteria. One challenge in coming up with global regulatory approaches is that it's uncertain how many devices will be in any side of the modifications that may be needed to achieve standardization. As I just mentioned, creating a one-size-fits-all approach would be difficult, as the evidence needed to support a modification depends on how much the company needs to modify the assay in order to achieve standardization. However, we have some general concepts here. Per the quality system regulation and recommendations provided in the FDA guidances, manufacturers must conduct a robust risk assessment to guide their decisions regarding validation and verification activities to support the modification. We think it's reasonable to believe that the studies needed to support the change in standardization will be analytical in nature, and companies are very familiar with study design for those studies, which may include accuracy, precision, linearity, and maybe reference range studies. Regulatory-wise, a legal predicate is required in a 510K submission. However, the meta-comparison study with the claim predicate may or may not be necessary. For instance, manufacturers can also use a well-established reference method, like the one used in the standardization program, as a comparator method to establish accuracy of the standardized device. However, there may be a need to characterize the difference between the standardized and the non-standardized assay to support the information that will be provided to customers. This means that update to labeling will be also needed to address what the new performance is and what this means compared to the results of the non-standardized version. From FDA perspective, these things are doable, and we look forward to learning about other regulators' perspectives on this. I also want to say that FDA can certainly find review efficiencies to expedite the review of submission of newly standardized assays. If FDA, for example, is proactively informed of the standardization programs that are being established with regards to the criteria for standardization, methodology use, and performance of corresponding reference method, it will ease processing all the 510Ks that will use the corresponding program. Since it's reasonable to expect that submissions across the companies will have similar information, our staff will be able to quickly conduct review as they get acquainted with those. For submissions that are of high quality, meaning that they are complete, detailed, and performance is acceptable, it is possible to use our triage program, which cuts the review time for a traditional 510K down to 30 days. The special 510K pathway that also has a 30-day timeline for FDA review may be an option. This is specific for previously cleared devices that are modified in a way that doesn't change the intended use and there are well-established methods to conduct the verification and validation activities. Of course, FDA will need performance information on the reference method to help inform our regulatory decisions. So for the first standardized assay, this path may not be appropriate. Again, these are just some thoughts, and FDA wants to do its part to support the standardization process. We believe that companies are quite knowledgeable and experienced in conducting analytical studies that may be needed to support the modification of the assay. However, in the past, we have shared with different stakeholders other important concerns that will need to be addressed for us to be confident in our regulatory decision. For example, we want to know, once the standardization of the assay is established, how will the change in result tests will be communicated to the practicing medical community? And also what type of outreach program is foreseen to educate and inform clinicians and practitioners about the clinical significance of the difference in patient test results using standardized versus non-standardized assays. And that concludes my presentation. Thank you all for your attention, and I look forward to the discussions that will take place in December 2021.